Photomorphogenesis, before we touch on this photomorphogenesis, so I would like you to understand the concept here. So as a human being, you have sensory organs or sensory responses. Okay, for example, you have eye, you have ears, you have nose, and you have your mouth, and as well as your skin. So you have all your sensory organ in order to in order to respond to the environment. So how about plants? So they are, like I said before, plants is just to imagine yourself, your feet, your both feet are buried in the ground, so you cannot move anywhere. You stay there, and then there is no way you can run away from the hot. You can run away from the dark, or you feel scared of the darkness. You can run away. So there is no way you can do that. So as a plant, so once you look at the plant, so have a pity on them. Not really pity, have a love on them. I love that because they have so many special systems in order to respond to the environment. So this photomorphogenesis can be defined as the process of the plants grows and differentiate to optimize their space using light. So in other words, that this is the light-mediated changes in plant growth and development. So sensing the light in the environment is very important for the plant because for the competition and also survival. So this response is mediated by a different photoreceptors. Later on, we're going to look at the photoreceptors present on the photoreceptors. Some are important. They have a lot of photoreceptors. So for your lecture, I want to focus on those important for you to know. Okay. So what also? There are some terms that you need to differentiate when we when you learn about this photomorphogenesis. So you have to know what is photoreceptors, you have to know what is photoperiodism, photomorphogenesis, just now we said, and also phototropism. So all the photo here. So not the photo, the picture you take. So the photo here is the response to the light, photon molecules, the light, okay? So the red light changes the shape of phytochromes and can trigger the photomorphogenesis. We're going to look on this later. So the stems goes from the etiolated in the dark or PFR phytochrome far red light to unetiolated in light with phytochrome red light. So it's okay. This don't afraid about this PFR and PR. We are going to look at it slowly one by one. Okay. So next, the control of this morphogenesis by light is called as photomorphogenesis. And then there are, as I mentioned just now, that the response is controlled or mediated by a different photoreceptors. So there are, for example, here we have 4 photoreceptors. Number one is the phytochromes. So very important for you to know these phytochromes. It is listed in your CI. I guess you see your CI, so you have to know about this phytochrome. Apart than all the 4 photoreceptor, phytochrome is the one that very important for you to know. Okay. So phytochrome is a blue pigment, a blue-green pigment. So the second photoreceptors is the cytochrome. Ataupun the cytochrome is under the protein receptors of this later on we're going to see of these phototrophin receptors. They are under this group. Okay, and then you have UVB photoreceptors for the UV light, and also you have this photochlorophyllite, a pigment absorb red and blue light and become reduced to chlorophyll A. So from these 4 photoreceptors, the one that we are going to cover in this lecture today is this phytochromes, the one that absorbs the red light. So the color is blue. The phytochrome is some kind of protein. So their color is blue. However, they absorb red light. So this cytochrome, they absorb blue light. Okay, going to see later. Okay, going to see slowly one by one. Okay. So red light and blue light responses. So we mentioned just now the red or far red light. These two we're going to look also for the photoreceptor responsible for absorbing this red light or far red light is phytochromes. So for the blue light, the photoreceptors involved ataupun the photoreceptor, the specific photoreceptors that absorb this blue light is protein-based receptors, phototrophin, and so cryptochromes. So the red light, so we go first for the red light and also far red light responses. So here for the the red light and also far red light, that the photoreceptor or the pigments or the chromoproteins important here is the phytochrome. So this phytochrome, they control the flowering in plant. So they are found in some plant cell and has been proven to control plant development. So most importantly is this phytochrome exists in two inconvertible form or phases or important here. So we have one is the PR, which is phytochrome red light, and PFR form. The second form is PFR form, which is the phytochrome far red light. Again and again, so that you don't confuse. Okay, there are two types of phytochromes. So the changes are into different phytochrome different, it depends on the wavelength. So for the phytochrome red light, the wavelength is they will absorb the red light with a wavelength of 660 nanometer. Whereas for the PFR ataupun phytochrome far red light, they will absorb the far red light with wavelength of 720 nanometer. So okay, again, this PFR is less stable, meaning that they are in the active form because they are not stable. So they will change to PR. So PR, just now I mentioned they are the inactive form. So this one active, they are not inactive. So in that condition, they will change to the inactive form. So sometimes this PFR, they will stimulate flowering, and the sunlight, they have more red light, so they will be absorbed, and also they will convert themselves into the PR will be converted to PFR in the so that they will become active. Okay, we can say that at night, okay, we have less PFR because they are the active form. In the day, we have more PFR. Okay, so at night there is a there is more PR but less PFR. Okay, don't confuse yourself. It will me also when I say tend to confuse because there is a opposite reaction of it. So hopefully you can jot down somewhere that okay. Who else? Who knows? I might ask in your final of test. So when I give you two situation, for example, I give you a situation of full sun and also in the darkness or under the shade of a tree. So hopefully you know this reaction. We have this kind of reaction later on below. There is a figure, there is some sort of mechanism ataupun the figure of it. Okay, next. So this is just to show the wavelength here. So you know that PR is 660, so they were supposed to be around here. PFR is 720, is it? 720, 720, correct. So they're supposed to be around here. Okay. So next, how this phytochrome works. Okay, again, like I said just now, the exposure to red light will convert this chromoprotein ataupun you can say the pigment lah. You can say depends on you, or you can convert the chromoprotein atau you can say the phytochrome to the functional active form, which is we call as PFR. While in darkness or exposure to far red light, convert the chromophore to the inactive form, which is the phytochrome red light. So if you see the reaction is quite opposite and so vice versa. So don't confuse yourself. Please memorize about this. Okay. Okay, this is the view ataupun the call it as the can we call figure? No, the flow. Yes, the flow. So again, since this phytochrome is a type of protein, so of course the synthesis come from the amino acids. So they will synthesis these two types of phytochromes I mentioned just now, incompatible types of the form of phytochrome. Number one is the phytochrome red light, and so phytochrome far red light. So in the absorbance of in the condition of the sunlight where the red light is more, so this phytochrome red light will convert to phytochrome far red light. Right. Okay. In the period of the darkness ataupun under the shade of a tree, so the plants tend to convert the phytochrome PFR since they absorb far red light. They will convert to the phytochrome red light. Alright. So this is going to lead on the destruction if they are depends on the situation ataupun maybe there are some of the biological response. So plants grow towards sunlight because the red light from the sun convert the chromoprotein into the active form PFR, which trigger the plant growth. Yes, PFR is the active form. Once active, they can do everything. So they tend to they tend to fasten their growth, and so they will do all the activities, cell activities, so in the very active lah, in a very active way. So in shade ataupun plants in the darkness ataupun shade will slow their growth because of the absorbance of the far red light will convert the phytochrome to the phytochrome red light, and then since this is the inactive form, so they will inhibit ataupun they're going to slow the growth. Okay, together these two forms of phytochrome represent the phytochrome system. So next thing is we finish about this darkness and also the light, the dark and also the light of this the phytochrome on the darkness and also on the light condition. So next we look at the germination. This far red light and also red light, how does they respond? How does the plant, plant germination respond to these two, this kind of system, this phytochrome system? So red light, if you expose the seeds to the red light, they will induce the germination. So if you expose your seed to the far red light, your seed will not germinate. There is a picture of it. You can search later on. Lettuce seeds exposed to red light and so far red light. So there is another test. They also did some tests on it. So the red light, they treated the seeds with red light, and then followed by the far red light, and then they bring the seed and then expose them to the red light, and the germination tend to be considerable. The germination, not all are germinated, and also not all are germinated fastly, but still they are giving a promising result of germination. Okay, another case is when we expose the seed to the red light, and then followed by then you take out and then expose them to the far red light, and then you take out back red light, and also and then lastly you expose the far red light, they will result on no germination ataupun low germination. So in that case, you can see the pattern here is once you expose them, the last step, if the last step there is the seeds are exposed to the red light, so there is a germination. If you stop your exposure of the seed to the far red light, meaning that no germination. Okay, those seeds are not rooted in the ground will get exposed to the red light, and this signal germination. This is why, and why. In certain situation, you cannot plant your seed very deep down the ground because they cannot absorb this red light. So they only absorb red light, far red light. So far red light, your germination tend to be low lah. Maybe if you, this is what I teach my final year project student. Don't put your seat too depth inside. Maybe more than 5 centimeter down. So your seat, it will take time for your seat to come up. After compared then if you plant your seed just at the top, maybe just tip a little bit, maybe just 0.5 inches. Still there is some part of them coming up. So inshallah they will germinate successfully. All right. Another thing is photoperiodism. So this photoperiodism is the growth of the plants according to day or night. So the length of night period plays a major role in determining which wavelength will be effective. Phytochrome pigment tend to revert to phytochrome during long period of darkness. Does the length of exposure to light affect how long the plant persists from to form the phytochromes? Okay. So this photoperiodism, usually we look it at the short day and long day plant. So okay, so this is not really happen to our country, so we can observe it in most of country overseas. So the day length perception, and then this one I want you to just read, and for your information, for us in Malaysia we are considering the day and also the night, the meaning the sunlight and also the darkness. So we are not considering the period of the short the day and also the night. Oh, all right. Next we move to the blue light here. All right. Finish about the red light and so on. So we move to the photoreceptors that contribute that are responsible for absorbing the blue light. So there are protein-based receptors, there are phototropin, and also there are cryptochromes. So these three groups, they sense a blue light wavelength. So for example, phototropin, they sense blue light to control a leaf opening and closing, stomata movement, and also opening of stomata. So when this phototropin activated by the blue light auxin, there is another things about this blue light. The auxin will accumulate on the shaded side of the plant, triggering the elongation stem and phototropism. Maybe if it's on the shade side, so they tend to elongate to the light. So if you're wondering what kind of hormone taken place there, it is auxin. So meaning that this phototropism works with the apical meristem, so we can control the development. So circadian clock is the endogenous timekeeper that keep plant responses to synchronous with the environment. So this rhythm of this circadian must continue to run in absence or external input. It must be 24 hours in duration. So it must be reset and trained or to determine or modify the phase or period of this rhythm by the light cycle. So they can compensate for the temperature differences. So again, the one that set this circadian cycle is blue receptors, which is this cryptochromes. All right, I guess that's all for today's lecture.